Okay, this is an introduction to the human immune system. It can be quite complicated, and we're going to try and make some sense of it and simplify it a little bit. First of all, let's look at the players in the immune system. The players in the immune system are the white cells. They all come from a stem cell and they develop into these various cells. Firstly, let's look at the lymphocytes, the T lymphocyte, B lymphocyte, and natural killer cells. We'll be talking about in a bit more detail, very important in the specific immune system. Eosinophils. Eosinophils are cells which are used in parasite infections. They usually target pathogens that other cells can't phagocytize, and they have killer granules which kill parasites. Neutrophils, they are the most plentiful in the blood. They are the first responders, and their job is to phagocytize debris and to secrete chemokines. Basophils are very similar to mast cells, and they both secrete histamine and heparin, and they are very important in the inflammatory response, which we will go into later, and they also come into allergy. Monocytes produce macrophages, which are the largest of the cells, and they ingest debris and pathogens and also have an important role to play. This slide shows the relative abundance of the various white cells, and the mnemonic "never let monkeys eat bananas." Most medical students know this, just to remind us which are the most plentiful in descending order: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Phagocytes come from the Greek, meaning that they eat and devour. They are cells that eat and devour debris. They have receptors on their cell membrane that detect foreign material. And the main phagocytes are neutrophils, macrophages, and dendritic cells. Let's have a look at the primary organ involved in the immune system, the lymphoid organs. The primary lymphoid organs are the thymus and the bone marrow. Red bone marrow is where all blood cells are produced. It is also the site of maturation of B lymphocytes. And B lymphocytes are named B because of bone marrow, or strictly speaking, they were first discovered in the bursa of Fabricius in birds. The thymus is critical to human immunity. Without a thymus, a person has no ability to reject foreign substances. The blood lymphocyte level will be low, and the immune response will be weak. So immature lymphocytes travel to the thymus, where they are kind of tested, and they put through two tests. One is, do they react to self? If they do, they are killed. And can they react to foreign antigens? And if they can't, they are killed. Only 5% of the T lymphocytes pass this test and leave the thymus. Alright, the secondary lymphoid organs, this is where lymphocytes find and bind to antigens, and an adaptive immune response is started. And this is where specific lymphocytes are activated and produced. Okay, let's have a look at the spleen. The spleen holds a reservoir of blood and destroys all the red cells. And it's just like a large lymph node that services the gut-associated lymphoid tissue. The tonsils and Peyer's patches are just really glorified lymph nodes. And lymph nodes themselves are small, oval structured, and they're located along all the lymph vessels throughout the body, and they're about 500 to 700 in the average human body. They are filled with lymphocytes, and this is where pathogens are brought to initiate the specific immune system. This slide shows the clever circulation of lymph tissue. So blood circulates out of the arteries and back into the veins, but over here about 20 liters of fluid is extravasated into the tissues every day. Of that 20 liters, 17 liters are taken back up into the vascular system, but the other 3 liters are drained via the lymph channels, lymph nodes, into the left innominate vein and the right subclavian vein. So what that does is it keeps lymph tissue all over the body. Lymph tissue rich in phagocytes, rich in lymphocytes, sort of patrolling every tissue in the body looking for problems. Okay, let's look at the 2 types of immunity. The first line immunity is called innate immunity. It's rapid, it's not selective, and it creates time for the specific immune system to set in. It consists of physical and chemical barriers, and it also has a strong cellular stroke chemical defense. Second line is the specific immunity, which is also called adaptive and sometimes called acquired. It's a slow response but highly selective and effective. There are 2 types of specific immunity: the humoral response, which is an antibody mediated immunity, and the B lymphocytes are involved in this. There's also a cell mediated cytotoxic system, and the T lymphocytes mediate this type of immunity. Okay, so let's look at the immune system, the innate immune system. Firstly, we get all the physical barriers. The skin contains keratin, which is a resistant type of protein. Other areas, especially openings, are protected by sticky mucus and mucous membranes, which trap pathogens. The nose and ears have hairs, and the respiratory tract has cilia. The skin and mucous membranes also create a chemical environment that protects the human body. Skin is acidic, and mucous membranes contain enzymes that break down pathogens. The stomach has a pH of 2, which is very unfriendly towards foreign bacteria. Then, of course, we have the normal commensals, of which there are many all over our body, and they outcompete pathogens for the nutrients that are available. Okay, so the internal defenses of the innate immune system, we're going to look at each of these in turn. Firstly, the inflammatory response, then the complement proteins, then phagocytic cells, and the natural killer cells. Okay, the inflammatory response has been, the signs of this have been known for a long time, and they were first documented by Roman Celsus in 38 BC in Latin, and we're all familiar with pain, heat, redness, swelling, and loss of function. And here's a picture of an inflammatory response. The white blood cells involved in the innate, virtually all of them, although the one circled in red, the lymphocytes, although they start coming into the area, they are priming themselves for the specific immune response. All the other cells, the granulocytes, neutrophils, eosinophils, basophils, the agranulocytes, the lymphocytes and monocytes, they all come into the area and have a job to do. Right, let's look at the flow chart of the inflammatory response. Imagine you are stabbed by an infected knife and pathogens are introduced. So the first thing that happens is that the mast cells and the basophils react to this invasion by releasing histamine and many other chemicals. Okay, this is the flow chart of the inflammatory response, and let's look at what might happen if you cut yourself with an infected knife. The first thing that happens is that the resident mast cells, which have a pattern recognition receptor, recognizes the PAMPs, the pathogen-associated molecular patterns. For example, like a virus, if there's a double-stranded DNA floating around, the cells know that it shouldn't be in the interstitium and it must be foreign. The mast cells release many pro-inflammatory mediators, mainly histamine, bradykinin, serotonin, leukotrienes, prostaglandins, and others. So what happens is that histamine dilates blood vessels and increases the permeability of capillaries. This brings sort of a lot of blood to the area, so it gets red and hot, and the increased permeability allows plasma and all the white cells to seep out of the capillaries and get to the area, causing swelling. So all of the white cells get to the area, and the neutrophils, because they're the most plentiful, are the first responders, and they can each phagocytize about 20 bacteria before they die. Eosinophils, as I mentioned, are specialized in fighting parasites and probably wouldn't be involved too much in bacterial or viral infection. Basophils are very similar to mast cells, and they secrete more histamine and heparin and other chemokines that are needed in the inflammatory response. Agranulocytes, the monocytes, which are the precursors of the macrophages, are the larger cells, and they can phagocytize about 100 bacteria each. The macrophages and dendritic cells, although relatively not plentiful, are important for presenting the antigen epitope to the specific immune system, bringing T and B lymphocytes into the picture. But that's a few days later. The innate response happens within a matter of hours. The complement system is part of the innate immune system, but it can also be called upon by the specific immune system to aid in that aspect of immunity. Complement consists of a number of small proteins, most of which are manufactured in the liver. It accounts for about 10% of the globulin fraction in the blood. And there are three different types of stimuli that trigger complement, which we won't go into now, but they all have in common that they take an inactive circulating protein and cleave it into an active protein that can actually do something. And what can it do? It can opsonize or coat the antigens, which mark them for phagocytosis, and the C3b is the component of complement that is most active in doing this. They can serve as inflammation chemokines, which attract more macrophages and neutrophils, and they can be directly cytotoxic by using the MAC attack, which is when a whole lot of these little complements develop into a spear and they kill cells, and C5b to 9 is involved in that. The last thing they can do is they can agglutinate the pathogens. In other words, they herd them together so that they can be killed in mass. Phagocytosis is activated by these molecular patterns that are found on pathogens, and phagocytes have these special receptors which recognize these PAMPs, as they are called. And white cells are amoebic-like, and they can chase foreign material around, driven by chemokines. Opsonins such as C3b and antibodies can act as attachments which aid the phagocytes in doing their job. Okay, let's look at two videos related to phagocytosis. The first one shows an opsonized bacterium being traced by a macrophage. It's a electron microscopy video, so let's have a look.
You can see that poor little bacteria there. Chase, 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 running around, running around. The other cells. And then pretty soon here, he's going to engulf it. Oh, there he goes. Isn't that cool? I mean...
Okay. The second video shows a researcher who's isolated some chemokines, and this is his own neutrophil from his blood, and he puts the chemokine near the neutrophil, and let's see what happens.
Video for you, and what you will see is one of our UCSF researchers who is using his own neutrophil and tempting it with one of these chemokines. And here you can see wherever he moves that pipette, the neutrophils go in that direction. So he's releasing these little sniffers, this bad smell that comes from the bacteria, essentially. You see how it goes right towards the pipette, right where he's releasing these little chemicals. So that's one main mechanism as to how these immune cells know where to go and how to sort of chase after those bacteria.
Okay, let's take a look at the natural killer cells, the NKTs. They are cytotoxic lymphocytes, which are critical to the innate immune system. They can detect pathogens that do not have antibodies. They come from the same progenitor as the B and T lymphocytes, but they're much larger and they contain deadly granules which kill cells. They are completely separate from the cytotoxic T lymphocytes, which kill by recognizing a specific antibody. And recently it's been shown that they also have a role in the specific immunity and in preventing cancer, and they are the subject of intense research, these interesting little cells. They kill cells with their granules, which contain perforins and granzymes. So they perforate the membrane and inject their granzymes, which totally dissolve the cell. And this is important because if they last a viral cell, the live viruses would be released. They kill the cell completely. NKTs represent about 10% of lymphocytes. Cytotoxic T cells need a MHC stroke antigen complex to kill, but these NKTs kill in a different way. They have two types of receptors on their cell surface. The first one detects MHC, and if they detect MHC, they say the cell is a self cell and the NKT is inhibited from killing the cell. They also detect activity ligands, which are found on most sick cells, and this receptor says kill the cell. So killing is done by a vote of which of these two receptors predominate. And just like T cells, they perforate and inject granzymes to kill the cell by apoptosis. Okay, the specific immune system, also known as the adaptive or acquired immune system. The specific immune system targets antigens on a pathogen, and this function is performed by the B and the T lymphocytes. These B and T cells recognize antigen because they have specific receptors on the surface of the B and T lymphocyte that exactly fit the antigen that they're looking at. The body doesn't know ahead of time what the antigen is going to look like, so it makes many different kinds of antibodies on the various B and T cells. So the average human is going to be exposed to about 1 million of these antigens in their lifetime, and there are many more in the human body that it is capable of responding to. Now, B cells are responsible for the antibody-mediated immunity, which is also called humoral immunity. And antibodies don't kill the pathogens directly, but they sort of attach to them and mark them for destruction. Antibodies bound to viruses, for example, prevent them from infecting cells. Antibodies can act as an opsonin, antibodies can neutralize a toxin, and they work best for free extracellular pathogens. Okay, so an antigen enters the body and is phagocytized by the macrophages or dendritic cells. And inside these cells, in this phagosome, the antigen is attached to the MHC type 2 complex, and it is presented on the surface of the cell to the T and the B cells. And these cells that do this and have an MH type 2 complex to do this are called APCs, antigen presenting cells, and these are professional antigen presenting cells. Okay, so the MAC activates the helper T cells and the B cells, and they move from being naive cells to being specific T and B cells with the antibody specific for the antigen that is being presented. B cells are activated directly by antigens. They're also activated by these macrophages, but the main way that they are activated is by the T helper cells. And the B cell system cannot work properly without T helper cells. Once we have these specific B cells with antibodies for the antigen, they start cloning themselves into plasma cells, and plasma cells are antibody factories. They secrete many antibodies, and they have antibodies both floating freely and on their cell surface to counteract the antigen. Some of these B cells become memory B cells, which live for a long time in the bone marrow and elsewhere in case there is a reinfection sometime down the line. Cell-mediated immune response, or cytotoxic immune response, is the job of the T cells. Again, the macrophage presents the epitope of the antigen to the naive T cell, which becomes a specific helper cell specific to that antigen. The Th2 cells stimulate the B cells, and the Th1 cells stimulate cytotoxic T cells to kill the antigen. Cytotoxic T cells are also stimulated by MH1 complex. MH1 is present in all nucleated cells in the body, and therefore there is some direct stimulation of the cytotoxic T cells. And these cytotoxic T cells kill in the same way as the other killer cells with perforins and granzymes. Okay, there are also T helper cells, and cytotoxic T cells also become memory cells in case there is a reinfection sometime in the future. At some later time, if we are infected with the same pathogen, the memory B and T cells are quickly activated, nipping the new infection in the bud. This is a slide to show how long the response takes the first time we are infected. You can see we've only got a decent antibody response and cell-mediated response 2 weeks after the antigen is injected. If the same antigen happens again, you can see how quickly, within a day or two, we have a much stronger and quicker response. And this is how immunity occurs after an infection. This is the full blood count of a young girl who had a tonsillectomy and bled 9 days later. And we can see from the leukocyte count that the total leukocyte count is very high, the neutrophils are high, and the lymphocytes are high. And this indicates to me that both the innate and the specific immune systems have had time to start developing.